Second lecture to Chapter 7's Photosynthesis series. In this lecture, we focus on the Calvin cycle, or otherwise known as the dark reactions. Let's do a quick review. Remember that the light reactions require a couple of things from the environment. That would include photons, represented by the light here. Remember that photons are wavelengths with specific amounts of energy. Which one of these has a greater amount of energy? If you said this one, you are right. Remember that the longer the wavelength, the weaker the energy. The shorter the wavelength, the greater the energy. The second thing needed from the environment is water. Do you remember what water contributes to the light reactions? If you said it's a source of electrons, you would be correct. Remember that the electrons are what we are tracking the entire time in the light reactions. We also need NADP, and we need ADP and phosphates. The NADP picks up the electrons that were donated by water. And when NADP picks up the electrons, see the electrons here, they go on to form NADPH. When ADP and phosphate go through the light reactions, remember in the ATP synthase, that's how ATP is generated by the plant. In fact, ATP and NADPH are really the important products, although we appreciate the production of oxygen because we rely on it for what we will learn will be respiration. We'll cover that in Chapter 6. But the ATP and the NADPH are important because they feed the Calvin cycle, which is the topic of this lecture. So with ATP, with NADPH, and CO2, specifically 3 CO2 molecules, a plant can go ahead and produce this sugar right here, G3P, glyceraldehyde 3-phosphate. But we can just say G3P. All right, so we need 3 CO2s, we need ATP, a source of energy, and we need NADPH, a source of electrons. Let's do a little review on those electrons. What happened to NADP when it picked up electrons? If you said it was reduced, you are correct. NADPH is reduced. How do we know that if we did not see the electrons? Well, NADP up here has no hydrogen, and all of a sudden it appears down here as NADP with an H. Remember that picking up electrons means you are reduced, and if you don't see electrons, picking up hydrogens is another way of determining that you've been reduced. Let's see what happens to the NADPH when it enters the Calvin cycle. It drops off its electrons and leaves once again as NADP+. What that means is that NADPH lost its hydrogen. And so what does that mean to us in terms of oxidation and reduction? It means that NADPH was oxidized. It no longer has that hydrogen. Also, what happened to ATP? ATP went into the Calvin cycle and it leaves as ADP with a phosphate. That means ATP broke down. Is that endergonic or exergonic? If you said it's exergonic, you are correct. Is that dehydration synthesis or is that hydrolysis? If you said that was hydrolysis, you are correct. Okay, so that's a quick review of some of the concepts that we learned from the previous chapter, Chapter 5, and also in this chapter on oxidation and reduction. Okay, so what exactly do I want you to know for the Calvin cycle? Well, I need you to know what the reactants are. And we covered those reactants in the first of the two lectures on the light reactions. If you recall, I gave you an outline and suggested that you should put these in some sort of flashcards. So the reactants, or the things that are required to get the Calvin cycle to produce G3P, are CO2, ATP, and NADPH. And again, the product is G3P. But remember also that in the course of donating its energy, ATP will be converted to ADP and phosphate. And remember also that NADPH will donate its electrons to become, once again, NADP with a plus. All right. So I need you to know where the Calvin cycle takes place. That's in the stroma of the chloroplast. I need you to know what the reactants are, CO2, ATP, and ADPH. And I need you to know what the products are. The products are, again, G3P, ADP with phosphate, and NADP plus. So how many things went in? Overall, 1, 2, 3 things went in. How many things came out? Overall, 1, 2, 3 things went out. I'm just going to count ADP and phosphate as collectively the second of 3 things that come out. All right, we can also see it here. This is a more detailed representation than this right here. So how many CO2s go in? 3. How many ATPs go in? 6. How many NADPHs? 6. And in fact, more ATP is required later on. How many G3Ps come out? 1. Okay, in a biomajors course, we would go through all of the individual steps, but I don't think that's entirely necessary here. We can summarize the steps. There's carbon fixation, and what this means is that you've trapped carbon, and we trap it in the form of CO2. So carbon fixation really is trapping CO2. Again, how many? We need 3 of those. Then there's an energy consumption step, or energy requiring step, and that's what we see here. ATP is required. And there's also a redox moment. That's this part where NADPH is dropping off electrons to become NADP plus. So if NADPH was oxidized, who was reduced? Well, that would be the organic molecules that we are creating here. And remember that you cannot have oxidation without reduction, or you cannot have reduction without oxidation. And that's why we say simply redox. And then there is also a release of G3P. And the Calvin cycle is a cycle, and that means that we regenerate something. We regenerate, in this case, a molecule called ribulose bisphosphate, or we can just call it RUBP. That's over here. It's a 5-carbon molecule. Okay. I would really invest my efforts here. I need you to know where the Calvin cycle takes place, and I need you to know what the reactants are and what the products are. There are 3 reactants, there are 3 products. All of this is happening through a series of enzymatic reactions, all taking place in the stroma, the liquid inside the chloroplast. All right, so in summary, there was a carbon fixation step. There was reduction of organic molecules, but that came at the expense of oxidation of NADPH. So this is the molecule providing the electrons in order to reduce what will eventually be sugars. sugar is called G3P. And then there, again, the regeneration of ribulose bisphosphate. So using carbon from CO2, electrons from NADPH, energy from ATP, the cycle makes G3P. That's the goal. Eventually, glucose is made. How? Well, you take G3P, that's a 3-carbon molecule. You add another G3P to that 3-carbon molecule. The end result is glucose. Well, let's go back. Glucose. And 3 carbons plus 3 carbons gives you the 6-carbon sugar we call glucose. All right, let's talk about an issue with photosynthesis that occurs under hot, dry conditions. And for this, I want to kind of review the structure of the leaf, the anatomy of the leaf. Remember that there's an upper epidermis? We covered this in the first lecture on the light reactions. That's this layer of cells that I've just drawn, kind of laid down flat. And then there's a group of cells that are tightly packed together at the very top. Do you remember the name of that group of cells? That would be the palisade mesophyll. And then there's a group of loosely organized cells. And you remember the name of that group? That's the spongy mesophyll. And below that, another group of epidermal cells. But this one has the 2 guard cells that form the stoma. Again, remember it's made up of 2 guard cells. All right. This story is about the stoma and what happens to it under hot, dry conditions. Remember that the stoma is the avenue that allows the entry of CO2 so that it can get to where it's needed, the palisade mesophyll, which is doing the bulk of photosynthesis. Remember that the stoma is also the way oxygen produced from photosynthesis will escape. Do you remember the photosynthetic equation? 6 CO2s plus 6 water molecules will give you C6, H12O6, and 6 oxygen molecules. So this story is about these 2 gases. The CO2 that should be going in, the oxygen that should be coming out. Under hot, dry conditions, the stomate closes. So imagine what happens if CO2 cannot enter and if oxygen cannot escape. Over time, what is the internal environment like inside the space of the leaf? Well, over time, whatever CO2 was in the leaf, say in these air spaces, whatever CO2 was there, that CO2 was consumed by the Calvin cycle, like we saw in the earlier slides. This means that CO2 levels are dropping. And what's happened to the oxygen concentration? Well, the palisade mesophyll is still photosynthesizing, so they're producing oxygen. So oxygen levels increase. And my point is that inside the leaf, CO2 levels are low, oxygen levels are high. And again, when does this happen? Under hot, dry conditions. Okay. All right, so what? What's the big deal with that? Well, the big deal is this. There's an enzyme called rubisco. All right, and you might be wondering, well, why is that an enzyme? Shouldn't it end in ACE? It does, actually. Here's the real name of the enzyme. This is the acronym, rubisco. But here's the full name. It's ribulose bisphosphate carboxylase oxygenase. Wow, it ends in ACE twice, in fact. Why is that? It turns out that rubisco is a very ancient enzyme. Think like old software versions, 1.0 versions, as opposed to like the more refined versions of software, version 8.0, 10.0, whatever. Rubisco is an ancient enzyme, and it's got several issues. The most important issue is that it cannot tell the difference between CO2 and oxygen. Do you remember that one of the properties of enzymes is that they are highly specific? Well, this enzyme, rubisco, isn't highly specific. It cannot tell the difference between CO2 and oxygen. What is it that the Calvin cycle requires of rubisco? It requires that it trap CO2. Rubisco is the enzyme that does carbon fixation. This is the one that grabs the CO2 from the atmosphere so that you can create a 3-carbon sugar, G3P. The problem is that rubisco cannot tell the difference between CO2 and oxygen. This is not normally an issue because normally there's a lot of CO2 and oxygen is in low amounts. And that's because the stomates are open, allowing CO2 in, allowing oxygen out, right? CO2 goes in, oxygen comes out. But what happens under hot, dry conditions? The stomates close, and they close because they don't want to lose precious water. They don't want to dehydrate. The leaves want to stay hydrated. So they close the stomates to prevent dehydration. As a result, no CO2 is coming in, and oxygen is not coming out. And so the internal environment becomes high CO2, low oxygen. And now rubisco has an issue. It can't tell the difference between either one. So rather than trapping CO2, it traps oxygen because oxygen is just more abundant than CO2. And as a result, the plant does not make a 3-carbon sugar. Instead, it makes a 2-carbon molecule. And when it does that, we describe it as photorespiration. Oops. All right, I wrote it up here. It means that during photosynthesis, you're trapping oxygen, which is a respiring type of step. Okay, so what kind of plants have this issue? It turns out that it's the majority of plants. So you've experienced this. What happens under hot, dry conditions if you don't provide enough water to your plants? They show signs of dehydration. They will not produce fruits or vegetables if that's the kind of plant we're talking about. They may not even flower if it's a decorative flowering plant. Photorespiration leads to poor production of sugars. Poor production of sugars leads to poor growth of a plant and potentially death. So let's take a look at some visuals of how some plants get around this issue. Because we do know that there are plants that survive perfectly well in hot, dry environments. You're probably thinking of things like cacti. right? Desert plants. So some plants have figured out a way around this issue of photorespiration. All right, so first, what are the types of plants that have issues of photorespiration? They're called C3 plants. Why do we call them C3 plants? Because when they trap CO2, the first thing that is produced is a 3-carbon molecule. All right, let's go back in our slide presentation and find that. Okay, here's CO2 being trapped, and how big is the chain of carbons that is produced after trapping, after fixing CO2? It's 1, 2, 3 carbons long. Most plants are C3 plants because they produce a 3-carbon molecule as soon after trapping CO2. But what happens under photorespiration? Rubisco, the flawed enzyme, traps what? Oxygen instead. So you don't make a 3-carbon molecule, instead you make a 2-carbon molecule, later broken down into CO2 and water. All right. Still, despite the fact that Rubisco is a flawed enzyme, it is arguably one of the most important enzymes on the planet. And how do we know this? Rubisco is the most abundant protein on the planet. In fact, Rubisco is so terrible at its job, trapping CO2, but so vital because it traps CO2, that the plant has no choice but to make tons of Rubisco. Because it turns out that Rubisco is also slow as an enzyme. Remember that enzymes are supposed to be fast? That's how they speed up reactions. Rubisco is slow. Something like a few reactions in a couple seconds. This is, again, a very ancient enzyme. So it's not the most specific, and it's not the fastest. But it's so vital because it produces sugar. So we need to have tons of it, and a plant just makes an enormous amount of it. As a result, because there's so many plants relying on Rubisco, every plant relies on Rubisco, and there's so many plants around the planet, that Rubisco becomes the most abundant protein on the planet. It's pretty amazing. Okay, so let's talk about the plants that get around the issue of photorespiration. They are called C4 plants. And I think you can guess why, right? The logic is consistent here. It's because when you trap CO2, these plants make what kind of compound? 4-carbon compound. There's two types of, or there's a subtype of C4 plant. That's called a CAM plant. It stands for crustacean acid metabolism, but I think we can just get away with saying CAM. These are the ones that you associate with cacti and other succulents like jade plants, and there's some others that I'll show you in a second. All right, so most of the plants on the planet are C3s. They're the ones that are vulnerable to photorespiration. But there are new and improved plants that have figured out a way around it. They are called C4s and CAMs. And why is it that they do better? It's because they have a brand new enzyme called PEP carboxylase. And that enzyme is way more specific at trapping CO2 versus oxygen. So when the CO2 levels are low inside of a plant cell that is a C4 or a CAM, PEP carboxylase is able to grab just CO2 and avoids oxygen. So what is the difference between the C4 and the CAM? Well, the C4 uses 2 cells, 1, 2. The first cell traps the CO2. You can see it here, being trapped and made into a 4-carbon molecule. And then that 4-carbon molecule passes through a plasmodesmata. Do you remember that's a communicating junction between cells, plant cells? And it frees up the CO2 and almost spoon feeds it to rubisco so that rubisco only ever sees CO2. And that reduces the potential of photorespiration. So trap it in one cell using something like PEP carboxylase, and then move the 4-carbon molecule across from one cell to another and drop off the CO2 literally in the mouth of rubisco so that rubisco never ever sees oxygen, it only sees CO2. But still, these plants, C4s and CAMs, they still rely on rubisco. All right, how does a CAM plant do it? The CAM plant only opens up the stomates at night. That's when it traps CO2 into a 4-carbon compound. At night, it makes sense because at night it's cooler. It's not hot. It still might be dry, but at least you're not dehydrating because of heat. The CO2 then is released during the day from the 4-carbon molecules. In fact, these 4-carbon molecules are stored in the central vacuole of these plants. And during the day, the central vacuole releases the 4-carbon molecule. It breaks down into CO2, and it gets spoon-fed into rubisco's mouth so that rubisco only ever sees CO2, and the 3-carbon sugars are produced. All right, so I want you to know what photorespiration is. I want you to know what kind of plants have issues with it. I want you to know what the solution is and who has that solution. The solution is PEP carboxylase, and it's C4s and CAM plants that are successful because of that PEP carboxylase. So let's look at examples of these plants. Here are the C4 plants. Corn. This is why corn is so important today. Corn is not only a staple crop for food, but it's also the kind of plant that can be used to make things like ethanol, a fuel, right, to power up engines. And with global climate change, corn becomes increasingly more important. We think that C4s like corn are going to be the sort of plant that survives and thrives in this changing climate that we have, this warming climate. Take a guess as to what this guy might be right here. This is sugarcane. It's also a C4 plant. It also is going to thrive in this hot environment. And then I'm sure you've seen this one, right? Nobody likes this one. This is crabgrass, right? If you have a beautiful front lawn made of tall fescue, that evergreen grass that needs a lot of water, but is also vulnerable to things like dog pee. If there's an area of that tall fescue that gets burnt by urine from your pet, it's not uncommon to find crabgrass immediately settling in that patch of exposed lawn and securing itself to that patch, grabbing a good foothold of it, and outcompeting your tall fescue. And that's because crabgrass is made for our Southern California, Inland Empire, hot, dry environments. It does really well, but we don't like it, and we prefer the tall fescues. It's softer on our feet. It looks prettier. All right, these are C4 plants. I want you to know these C4 plant examples for your second exam. Here are examples of CAM plants, the succulents. They include things like agaves, right? They include the cacti. This is opuntias. You can recognize a prickly pear. So this is prickly pear cacti. That's the fruit. Tunas in Spanish. Here's jade. I used to play with jade when I was a kid. We'd pluck jade leaves, play tic-tac-toe on the concrete. They're so juicy, they would just help us write on the concrete. And most students are surprised to learn that pineapples, although they're associated with tropical environments, pineapples are actually the sort of fruit that might grow in an arid sort of island. Not everything that's in a tropical region is going to be lush islands, right? Some islands in tropical regions are actually hot and dry, that there's just not enough moisture there. And pineapples do well in those environments. And obviously, they'll do even better if you provide them with water, right? So some tropical type of areas do well growing pineapples. Okay, I want you to know about pineapples, agaves, cacti, and jade as examples of cam plants. Let's move on. Let's talk about that issue of the changing climate, the global climate change that we're going through. Let's do a quick review of something you've heard about before, probably called the greenhouse effect. The greenhouse effect is a product of Earth's insulating gases, the gases that are part of our atmosphere. When solar energy strikes the planet, some of that energy radiates back out towards space. Fortunately for us, that heat energy that would otherwise escape into space is reflected back onto Earth by our atmospheric gases. And it's that radiating heat that is pushed back to the planet that warms our planet. The gases that help warm the planet, that help insulate the planet, are CO2, water vapor, things like methane, right? These are all insulating gases. They reflect the heat that would otherwise escape back to the Earth. So the Earth, despite the fact that it has a molten magma core, right, liquid core of essentially like melted rock, despite having that, it is not enough heat, or that heat is not going to be able to keep the planet warm on the surface. We rely on the sun's energy to keep the planet warm on the surface. And even that energy can bounce off of the surface of the Earth. Again, the atmospheric gases bring that heat energy back to the surface of the Earth, and they keep the surface of the Earth warm. Without our atmospheric gases, the Earth's surface would be below zero, and a lot of things would die. Earth's creatures would be different. Well, maybe not necessarily different, but there would be certainly fewer. There would be the sort of creatures that can survive in extreme cold weather, right? Okay, let's take a quick visual of that. So here's the sun's solar energy, the same sort of stuff that drives photosynthesis, but it leaves the planet in the form of heat energy. And again, the atmospheric gases here form like a blanket, and they redirect the heat back. And that's what keeps the surface of the Earth nice and warm. Let's talk about when we first learned that we had potential issues with CO2. Sometime in the 50s, technologies improved in such a way that we were able to get better estimates of the concentration of CO2 in the atmosphere. What you're looking at here is called the Keeling curve, and it shows that CO2 levels go up and down seasonally. So here's the data collected from Mauna Loa. This is in Hawaii, or the Hawaiian Islands. And you can see that CO2 levels go up, down, and up again. And if we look at the calendar, right, we can see that the rise and decline of CO2 concentrations seems to be seasonal. So take a guess. When is it, do you think, that CO2 levels go up? And when is it that CO2 levels go down? So CO2 levels are high when? Fall and winter. Why do you think? That's because that's when plants tend to go dormant, right? Deciduous trees drop their leaves. And that's because the days get shorter. So there's not enough sunlight to sustain photosynthetic growth. When do CO2 levels drop? That would be in the growth seasons, right? Spring and summer. This is when the days are longer and now plants are going to more efficiently produce sugars for growth. So this is what we learned. We learned that CO2 levels go up and down throughout the year. And we understood why. But how long were we recording? Well, it's 58, 59, 60. That's just a few years, right? In fact, it's just a couple of years and change. But we kept recording CO2 levels, and here's what we found. They're still going up and down overall, right? But what's the major trend here? CO2 levels still go up, down, up, down, up, down, but overall they're doing what? They're definitely climbing. CO2 levels are climbing hard. And now this is spread out over a much longer period of time. Okay, so if we understand that CO2 is one of the important gases that insulates the planet, and then we find that CO2 is increasing over time, I think you can put two and two together and deduce that, hey, this might be an issue, right? We're making our blanket thicker. Shouldn't that warm us up more efficiently anyway? Yeah, so that was the concern. But you might say, you know what? How do we know that these CO2 levels that are climbing are a product of human activities? What if these CO2 levels are climbing naturally as part of like a natural cycle that the planet has always gone through? How do we know it's us and not something that's just completely natural? It's only a small amount of time given the age of the Earth. Yeah, we're seeing CO2 levels go up, but are they going up because of human activity? Or are they going up because it's just a natural tendency or a natural cycle or a natural phenomenon that the planet has always gone through and we're just taking notice of it now? Well, maybe we should go back in time and see what the CO2 levels were. So how do we do that? How do we go back in time to determine what the CO2 concentrations were? We go to polar regions and we drill into those polar regions and we pull out ice cores. And those ice cores, frozen ice cores, have trapped air from a long time ago. We're going to learn about something called the law of superposition when we discuss evolution. But basically what it says is that the farther down you dig, the farther back in time you go. So if we dig polar ice regions deep, deep, deep down, we're going to take a glimpse as to what the atmospheric gases were like many, many years ago. So what do they look like? All right, here it is. If we went back close to 500,000 years, we would see that the CO2 levels are here. They're under 300 parts per million. And then what happens? CO2 levels decline. These are associated with ice ages. And then CO2 levels climb up again, right? roughly about 100,000 years. Then they come back down, go back up, close to another 100,000 years later, go down, come back up, but never really exceeding what? 300 parts per million. This is, again, 500,000 years. And again, the pattern continues, right? And then look at this. Whoa, where did that come from? Well, this is current time. This is now human civilization. And what we think has happened is that because of industrializations, because we were pulling trapped liquefied CO2 and petroleum out of the earth where it was stored safely, we burnt it, put it into the atmosphere, we increased the amount of CO2 in the atmosphere. So we're convinced that this is attributed to human activity and not to natural phenomena. In fact, recently, the CO2 levels have surpassed 400 parts per million. That hasn't happened in over 500,000 years. This is us. We're doing this. Okay, we'll talk more later in the semester about this, but what are the consequences of rising CO2 levels and thickening the insulating blanket in our atmosphere? Well, some of these are predictions and some of these are actual events we are monitoring, recording today, like melting of polar ice caps. And the problem with that is that the melting of these ice caps means that we're going to raise sea levels. Raising sea levels means that coastal communities are going to be underwater, right? That's rising sea levels. It also means that we're going to experience extreme weather patterns. If you insulate the planet, we're going to throw off the rhythm of our weather patterns. And we have noticed some pretty extreme weather conditions. Some places are going to experience droughts as the planet warms in some areas. They're going to experience droughts. California is experiencing a drought, right? We're asked to conserve water. We're just not getting enough snow, which is where Southern California tends to get its water from. We get them from our Sierras. So we can get all the rain, and we've had significant amount of rain a couple of winters now. But we're not dependent on that rainwater as a source of water for ourselves. We depend on snowpacks in mountains to get our water. We'll talk more about this later in the semester. We're also seeing increase in extinction rates. We'll talk more about mass extinctions also later in the semester. And we're also seeing the spread of tropical diseases like dengue fever, mostly because mosquitoes are thriving in this changing climate of ours. Okay, so global climate change is an issue. What can you do to help offset this climate change? You don't have to do a lot. There's plenty of us, and if each one of us does a little, our collective effort can reduce or bring back the CO2 levels to something that's more of the norm, given the data that we've seen. You can plant trees, right? You can do something about putting plants in the ground, as opposed to the overall slash and burn of forests that we're also seeing that is just precipitating this problem. Okay, let's save those conversations for later in the semester as well. Another global issue associated with photosynthesis that we ought to talk about is the ozone. So what is ozone? That's O3. It's weird, but it's three oxygens attached to each other. And we rely on ozone heavily for this reason. The sun doesn't just give us energy, it also gives us UV rays. And I think you all understand that UV rays are damaging to our skin. UV rays are associated with skin cancers, and this is why we use sun protection, right? So how does the ozone help prevent UV rays from reaching us? Well, when the UV strikes ozone, it breaks it up into oxygen and a free oxygen, right? And it's very reactive, this oxygen. So it'll just grab another oxygen, link up with it, and produce O3 once again. And this is a dynamic process. It's constantly breaking up and reforming, right? Another oxygen there. So they just recombine to form O3. So as long as we have ozone, we have protection against incoming UV rays. However, all through the 80s and earlier, we started relying on a chemical called a chlorofluorocarbon. We can just abbreviate here, CFCs. They are refrigerants. They are designed to compress gases into a liquid form in a canister and make them so pressurized that with a little valve, you can release a mist of that pressurized gas. Some of you from the 80s might remember Aquanet. We used to carry that in our backpacks back in the 80s. Sport these spiky hairdos. But it was also in other refrigerants. It was in our AC in our vehicles. It was the refrigerant in our refrigerators. But those CFCs, once released out of these containers, these pressurized containers, these CFCs would get into the atmosphere. And here's the problem with CFCs. Remember that UV strikes O3 and breaks it into oxygen and a free oxygen? Well, CFCs would bind to that oxygen, and when they bind to it, they won't let go to it. And by not letting go, we cannot regenerate what? O3. So we were putting so much CFCs in the atmosphere that we started noticing a thinning of the ozone layer that protects us from UV rays. And so we started doing something about it. So the CFCs thinned the ozone layer, and then we started documenting the thinning of that ozone layer. Agencies like the British Antarctic Survey, NASA, National Oceanic and Atmospheric Administration, all the scientists of these organizations documented the thinning of the ozone layer. And in response to that, a treaty addressing the Earth's environment was signed in 87. And over time, other nations came on board and followed protocols to reduce the CFCs that were used in a variety of refrigerants and aerosolized products. As a result, the ozone is coming back. So this is a win story, right? For the identifying of an environmental issue, implementing policies that then spread to various nations, and our collective efforts reduce the impact of this environmental concern. We hope that this is the sort of story that helps inspire something similar with global climate change. And many nations jumped on board. Unfortunately, recently, our U.S. Has jumped out of these treaties, unfortunately. All right, so global emissions of CFCs as a result of these policies are near zero now. But because the CFCs are stable, the ozone is still going to require some time before it fully heals, right? But UV rays are going to increase the potential of skin cancers, things like cataracts, could damage the photosynthetic abilities of crops that we rely on for food, and phytoplankton, that is essentially the energy producing organisms of the ocean. But we should also care about other organisms that would be affected by the thinning of the ozone layer, like the emperor penguin or fur seals that you find in places like Antarctica, right? We are intelligent beings that can do something about the environmental issues that we create ourselves, right? These creatures had no say in this matter. We ought to feel bad for them. This is our doing. All right, so we've covered photosynthesis. What's next? Well, we learned how to make organic molecules. We know where oxygen comes from. It's a product of photosynthesis. We're now going to take the sugars and the oxygen that were made by plants, and we're going to send them to the mitochondria, our mitochondria. Our goal is to make ATP, the energy currency of the cell. And when we make ATP, we create CO2 and water. And guess who likes that? Chloroplast. Plants do. So there's a really cool cycle between the chloroplast of plants and the mitochondria of the rest of us, the animal world.